Hello, how are you? We talked about oxidative stress on the in the last lecture, and I hope you have understood it. You cannot understand antioxidative mechanism unless and until you understand oxidative stress very properly. If you have some doubts, please go to that presentation, go to that video again and again, and try to follow it. We were talking about the GSH system also. And today we are going to discuss the antioxidative mechanisms or antioxidants. When we started our discussion on oxidative stress, we said that oxidative stress is defined as as an imbalance between pro-oxidants and antioxidants. It means antioxidants are already there in the cell, are already there in the system. Nature provided all organisms with these mechanisms to fight against those agents which cause oxidative stress. And those agents are the reactive species, those agents are the radicals, the free radicals. We have discussed about them. So, if we begin with this concept that it is an imbalance between the oxidants and antioxidants, we come across with this that all aerobic organisms need oxygen. We talked about that this is a paradox that oxygen is needed by all aerobic organisms, but oxygen is toxic. How much it is toxic? Only 2% remaining 2% oxygen which has not been used by the cell, that becomes toxic. 98% of the oxygen used or consumed by the aerobic organisms is used by through a cytochrome system through the respiratory chain of the mitochondria, but only 2% which has not been used becomes deleterious, becomes damaging, becomes poison. So we are concerned with only this 2% of the oxygen which was left in the cell. How this oxygen is uh, is destroyed. So nature provided to destroy it and its products, the subsequent products, with some enzymes. And these enzymes are called as antioxidant enzymes. These antioxidants are classified into two groups. The one group is the enzymatic oxidants, and another group is the non-enzymatic oxidants. If we talk about the first, let us discuss about the enzymatic oxidants which are already there in the cell, which are you see there is an antioxidative element, a protein and gene. When we combine these together, it becomes an antioxidative element. So there is an antioxidative element existing in the cell, and this antioxidative element is protecting the cell against these pro-oxidants and their products. So the first enzyme in enzymatic antioxidants is superoxide dismutase. Superoxide dismutase. It is very important. We call it as SOD. And the latest researches, they tell us that superoxide dismutase is not one enzyme, it is a family of enzymes, or you can say that there is a genetic polymorphism in superoxide dismutase. There may be, and moreover, there are different types of dismutases. There are some which need iron for catalysis. There are some which need manganese. There are some which needs copper. There are some which need zinc. So there are copper dependent, zinc dependent, manganese dependent, iron dependent superoxide dismutases. And their role is when the diatomic oxygen is produced, when free radical or superoxide anion is produced, the two anions they join together, and they with the help of dismutation expressed by the superoxide dismutase, they change into a less dangerous, which you call is less dangerous hydrogen peroxide plus oxygen molecule. This is how this superoxide dismutase is working. O2, and this is the hydrogen is added with the help of superoxide dismutase. It becomes H2O2 plus the oxygen. So fine, now we have hydrogen peroxide, and this is again very dangerous. So this will be dealt by something else. So the major function of the superoxide dismutase is to dismutate the oxy radical and produce hydrogen peroxide and oxygen. The second function of superoxide dismutase is that sometimes it happens that the nitric oxide is generated, and it comes from the endothelium, it comes from the Kupffer cells, it comes from many other cellular sources. So when the oxygen and superoxide anion and NO join together, or another very dangerous species is formed, and that dangerous species is peroxynitrile. Peroxynitrile. What does it mean? ONOO. This is peroxynitrile. This is the already the NO, you see, NO has a very important role to play in vasoconstriction. NO has got very important role to play in this cell. But sometimes when this NO is generated in excess, in the conditions, in toxicity conditions, then what will happen? Then superoxide anion and the NO will join together, and then there will be the formation of a peroxynitrile, ONOO, and then this ONOO will be dismutated with the help of superoxide dismutase, and we will get another peroxynitrous acid, and that peroxynitrous acid will be ONOOH. That will be ONOOH plus 2 hydroxyl radical, 2OH, OH radical, hydroxyl radical plus 2NO2 will be formed. So in this reaction again superoxide dismutase is playing a very important role. Superoxide dismutase quenches the superoxide anion. Superoxide dismutase quenches the peroxynitrile. Superoxide dismutase quenches the peroxynitrous acid. So in this way, superoxide dismutase plays a very important role in all aerobic organisms. The role of superoxide dismutase has been confirmed by using the superoxide dismutase null mice, that genetically a mice is produced where there is no superoxide dismutase, and when we abuse the rat or mice with some with pro-oxidants, then there is no superoxide dismutase, so the mice dies. So this is a very important enzyme for aerobic organisms. The another very important enzyme for aerobic organism is the glutathione peroxidase. We were talking about glutathione peroxidase in the last lecture also. Glutathione peroxidase, as the name suggests, is responsible for quenching hydrogen peroxide, is responsible for quenching all peroxides which are formed due to the metabolism of the pro-oxidants. Glutathione peroxidase was discovered by a person, Leopold Flohe. सुपरऑक्साइड डिस्म्यूटेज पर काम करने वाले लोग कौन? हेलमुथ साइज, बोवेरी, राडी, लेकिन एक फ्लोए नाम के एक जो साइंटिस्ट हुए, उन्होंने लियोपोल्ड फ्लोए, उन्होंने सबसे पहले ग्लूटाथियोन पेरोक्सीडेज को ढूंढ निकाला, और उन्होंने कहा जब हम ग्लूटाथियोन सिस्टम की बात करते हैं उस वक़्त जो पेरोक्साइड्स फॉर्म होते हैं, हाइड्रोजन पेरोक्साइड को पानी में बदलने वाला कौन? पानी में बदलने वाला जो एंजाइम है वो ग्लूटाथियोन पेरोक्सीडेज है, which is mostly present in mitochondria. Glutathione peroxidase, as we were talking about, it is again a family of enzyme GPX1, 2, blah 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 blah, and glutathione peroxidase. When we talk about glutathione peroxidase, we cannot ignore selenium, because we say that whether this glutathione peroxidase is selenium dependent or selenium independent. So we talk of selenome, or we talk of selenocysteine, and it becomes selenoproteome. सेलिनियम पे बहुत काम होता है, सेलिनियम पे बड़े बड़े रिव्यू लिखे जाते हैं, सेलिनियम का बायोलॉजिकल रोल क्या है, सेलिनियम सेल के अंदर क्या करता है, और जब सेलिनियम की बात करते हैं then we do talk of glutathione peroxidase, because selenium or selenocysteine complex works only with the help of an enzyme. That enzyme is called as glutathione peroxidase. The role of the glutathione peroxidase is very simple. It eats hydrogen peroxide and it gives rise to two molecules of water. H2O2 is changed into 2H2O with the help of this selenium, selenocysteine. So cysteine is an amino acid. Selenium joins with that amino acid, it becomes selenocysteines. Selenocysteines are very, very much important in pharmacology, in physiology, as well as in toxicology. So when we talk of glutathione peroxidase, we do talk of selenium dependent glutathione peroxidase, right? So whatever will be, you saw that in the first reaction superoxide dismutase के बाद में बना क्या था, हाइड्रोजन पेरोक्साइड. सो भाई उसको कोई डील करने वाला तो होगा, तो नेचर ने उसको डील करने के लिए आपको क्या एंजाइम दिया, वो एंजाइम दिया ग्लूटाथियोन पेरोक्सीडेज. ग्लूटाथियोन पेरोक्सीडेज की मदद से हाइड्रोजन पेरोक्साइड को पानी में बदल दिया गया, right? It was changed into water. Somebody else may also watch my lecture, so I am not using Hindi, I am using mainly English, so please pardon me for that. Now there is one more enzyme, glutathione peroxidase का काम क्या, glutathione peroxidase का काम to eat the peroxides. There is another one very important enzyme, third enzyme, that is catalase. उसका काम भी यही है, that is also functioning to scavenge, to remove the hydrogen peroxide, and with the help, but the products are different. There we are getting two molecules of water when glutathione peroxidase is working. When catalase is working, we are getting two molecules of water plus oxygen. O2 plus 2H2O. It is like this, 2H2O2 catalase, O2 plus 2H2O. So in this way we get oxygen as well as water. There we get only the water, two molecules of water. So this catalase plays a very important role as an antioxidant enzyme or as an antioxidant mechanism. I was fortunate enough to work with a person who is an authority on catalase, Professor Masana Ogata in Japan. He is an authority on catalase, and he wrote a very good review article, Human Catalase in Human Genetics. I can just tell you that those people who have no catalase enzyme in their blood, who have no catalase enzyme in their body, they are called as acatalasemic. And those people who are having a less amount of catalase in their body, they are called as hypocatalasemic. That is the now what will happen, there is a population in Japan. The male Japanese soldiers during World War II married some Thai and Korean ladies, and they got a genetic disorder, the genetic deficiency. And that genetic deficiency is called as acatalassemia. The children who produce, they have no catalase enzyme, so they are more prone to the toxicity of hydrogen peroxide. So catalase is playing a very important role. The next enzyme in this series is glutathione reductase. I talked to you in my previous lecture also that when GSH is consumed, so the oxidized glutathione, GSSG's responsibility is to generate or to replenish the lost amount of glutathione, reduced glutathione. And that is done with the help of an enzyme which we call as glutathione reductase. So glutathione reductase is also very important. They are all glutathione dependent enzymes. Glutathione transferase, glutathione peroxidase, glutathione reductase, and glutathione itself. These are, they all make a glutathione system, that GSH system that we discussed in the last lecture. And in addition to that, glutathione peroxidase, which is again having GPX1, 2, 3, 4, 5, alpha, beta, theta, like this, they have got the polymorphism, they exhibit polymorphism. So when there is polymorphism, the substrates are also different. So in this way, the glutathione reductase helps in generating the reduced glutathione. Another enzyme which is responsible, which works as a antioxidative enzyme is glucose 6-phosphate dehydrogenase. You should remember. First enzyme, superoxide dismutase. Second enzyme, glutathione peroxidase. Third enzyme, glutathione reductase. And the fourth enzyme is glucose 6-phosphate dehydrogenase. You see, it is a very important disorder in human population. 400 million people throughout the world do not have glucose 6-phosphate dehydrogenase in their blood. And the result is they suffer from hemolytic anemia. Hemolytic anemia is caused due to the deficiency of this enzyme glucose 6-phosphate dehydrogenase. Dehydrogenase protects against hemolytic anemia. Dehydrogenase protects against malaria. This is needed to protect a population against malaria. So it also helps us to protect the population from drug-induced anemia. Drug-induced anemia. So in this way, glucose 6-phosphate dehydrogenase is again a very important enzyme. Though antioxidative role of glucose 6-phosphatase dehydrogenase has not been established properly, or has not been studied properly, but the significance of those enzymes, SOD and GPX and GRD, glutathione reductase, and catalase, that has been very well established. We worked on a very interesting topic. We collected different animals and started studying the catalase activity in their blood samples and liver samples, and we correlated that with their feeding habits. And we found that those carnivorous animals which eat the rotten stuff, like snake eats the rat itself, and the rat has eaten so many rotten stuff, but the snake is able to survive. It is only because of the catalase, the H2O2, which has been produced as a product of this digestive activity, that was neutralized by catalase. Another very important study that we made sometimes back in acatalasemic mice, when we expose acatalasemic mice to carbon tetrachloride, we found that the glutathione peroxidase activity was high enough. It increased as an adaptation because there is no catalase. So the glutathione peroxidase has the responsibility to remove the hydrogen peroxide. So in this way, what do we find? That the nature has provided us with an inherent system, with an inbuilt system, along with the responsible genes and proteins and enzymes. Then this inherent or inbuilt system is protecting the cell, our cell, from the insults which are caused by the oxyradical, peroxyradical, hydrogen peroxide, hydroxyl OH radicals, and another radical, including the NO, nitric oxide. I tell you, O and NO join together and they make a peroxynitrile radical. So to fight against these oxidant products, the nature has given you an enzymatic system. One of your colleagues was asking, sir, if it is it is like this, then why can't we give the antioxidant from outside? This is an inbuilt system, right? When we are going to talk about the non-enzymatic antioxidants, they are not inbuilt. They are administered, or they have got a very important role to play. In my next lecture, you will listen to the non-enzymatic antioxidants. I think I am clear about these enzymes. In brief, I have discussed you their significance, the biological significance of antioxidative enzymes. Keep on studying, and we shall be discussing more. Thank you for listening.